O positive is the most common blood type globally, found in over a third of people. It lacks A and B antigens yet has the Rh factor on red blood cells, making it compatible with any Rh positive recipient. This exceptional compatibility makes O positive the foundation of emergency blood transfusion, letting doctors quickly stabilize patients in crisis situations. Medical systems rely on regular O positive donations to keep blood reserves stable. Its abundance ensures it remains a mainstay in hospital practice. Biologically, O-positive individuals present unique traits. Because they lack A and B antigens, there are fewer complications when exposed to foreign blood types and slightly lower susceptibility to specific diseases. In regions plagued by malaria, O blood notably improves survival since Plasmodium falciparum, the malaria parasite, clumps A and B cells more aggressively than O cells. This evolutionary protection reveals why O type is dominant in tropical and equatorial areas. However, O blood structure carries vulnerabilities too. The exposed H antigen, which stands in for the missing A and B markers, acts as a target for pathogens like cholera and norovirus, making O types more sensitive to severe gastrointestinal infections. O-positive individuals tend to have lower levels of proteins like von Willebrand factor, slowing their blood clotting rate, a protective factor against thrombosis, but a potential risk during injury or surgery. A-positive comes next, present in nearly 30% of the population. Its identity, the presence of A antigens plus the Rh factor. These surface proteins help the immune system recognize self from foreign cells, and people with A positive can receive blood from A positive and O positive donors, giving hospitals a broad supply pool. Its prevalence and compatibility make A positive transfusions easy to manage. Evolutionarily, A antigen carried certain advantages for early farming communities, increasing immune response against bacterial invaders in crowded settlements. This improved resistance helped type A populations survive the transition from hunting to agriculture. However, A antigen's immune robustness comes with drawbacks. Elevated immune activity can drive inflammation and increase the risk of arterial thrombosis or heart disease. Clear evidence of the trade-offs that shape blood types. B-positive blood is found in around 8 to 10% of donors. It is defined by B antigen and the Rh factor. People with B-positive can receive blood from B or O donors, regardless of Rh factor. B-positive blood is less common, especially in Western populations, so maintaining a reliable supply is harder in some regions. Genetic maps show B originated in South Asia and spread unevenly, peaking in Asia and the Middle East. B antigen supports adaptation to distinct diets and immune stressors. For example, B antigen discourages colonization by certain E. coli strains, but encourages others. And B-positive individuals process dairy and carbohydrates slightly differently, which traces back to ancient dietary patterns. AB-positive, the rarest positive blood type, is found in fewer than 5% of people. It combines A, B, and Rh antigens, making these individuals universal recipients. They can receive blood from any group. This flexibility is life-saving for complex transfusions, organ transplants, and emergencies. AB positive arose recently in evolutionary terms, likely as A and B populations intermingled within the last 2,000 to 3,000 years. With both antigen systems present, AB positive blood offers broad tolerance to foreign antigens, but increases clotting proteins and inflammation markers, raising the risk of vascular conditions. Their immune profile is balanced, yet it is not without medical trade-offs. O-negative is carried by just 7% of people. It lacks all primary antigens, allowing O-negative red cells to be transfused to any patient regardless of blood type and earning it the title of universal donor. In emergencies where blood typing cannot be determined, O-negative is used first, often saving lives. O-negative blood's rarity makes these donors essential for trauma care and disaster response, and blood banks prioritize this type in supply management. The scarcity of O-negative reflects genetic uniqueness. It's less common historically and in the modern world, as most populations are Rh positive. O-negative people share O-type benefits, such as malaria resistance, but also O's increased risk for gastrointestinal infections and the added risk from Rh incompatibility during pregnancy. Rh negative mothers carrying Rh positive babies may develop antibodies that damage fetal red cells, a risk now mitigated with modern medicine. Here is an interesting fact. People with O-negative blood can only receive blood from other O-negative donors. This is because O-negative blood has no A, B, or Rh antigens, meaning that introducing any other blood type would trigger an immune reaction. A-negative is present in just 2% of people. It displays A antigen, but lacks the Rh factor, narrowing compatibility options to A-negative or O-negative blood. The scarcity heightens the importance of each donation, especially in hospitals serving maternity wards and surgical centers. Historically, A-negative individuals benefited from resistance to bacterial infections in dense settlements, a trait tied to ancient agrarian societies. However, pregnancies involving Rh incompatibility posed serious risks before medical intervention. Modern management with Rh immunoglobulin injections now prevents maternal antibody development that can harm the fetus. B-negative, one of the world's rarest types, appears in only 1% of donors. It's defined by B antigen and no Rh factor, and compatible donors are so limited that blood centers tightly manage B-negative supply. B-negative is more common in Asian populations and rare in Western ones. Its spread likely arose in areas where B antigen and Rh negative mutations intersected. B-negative carriers are less prone to some bacterial infections, but more vulnerable to inflammatory reactions and certain digestive tract illnesses, reflecting finely tuned evolutionary trade-offs. AB negative is a recent and complex fusion from rare parentage, a genetic outlier, and key to understanding global blood type variation. Its mix of antigens and rarity highlight the deep evolutionary roots and medical relevance of blood inheritance. Blood types aren't limited to the ABO and Rh systems. Genetic inheritance underpins every type. Blood group antigens, a result of gene combinations, are inherited like eye color. Their frequencies vary by population and region. Some rare types, like RO, are more prevalent among Black heritage populations, and this shapes how blood banks manage supply and demand for different communities. Genetic diversity extends beyond ABO and Rh. New antigens, such as the ANWJ protein, further complicate transfusion matching, but can be life-saving for patients with rare disorders. Most people have ANWJ antigen, yet it can only be identified through advanced genomic screening in rare cases requiring highly specific donors. Tracking and identifying these subtypes help manage rare transfusion reactions and support the safety of patients with unusual genetic profiles. Disease resistance is heavily influenced by blood type. The ABO system's evolutionary roots lie partly in infection risk. Type O is less sensitive to severe malaria since the lack of A and B antigens prevents the parasite from clumping red cells, a major evolutionary advantage in tropical environments. However, O carriers face higher risks for cholera, mumps, and norovirus, since pathogens exploit the exposed H antigen. During major outbreaks, O types show higher mortality and more symptoms for these diseases. The A antigen provides robust defense against certain bacteria, such as Pseudomonas aeruginosa, but A types are more prone to smallpox and higher rates of stomach, ovarian, and colorectal cancer. For B types, Streptococcus pneumoniae and E. coli become less threatening, but susceptibility increases for gonorrhea, tuberculosis, and some viruses. B types also process carbohydrates and fats differently, a trait that served ancient farmers and herders consuming regional diets. Type AB merges the immune nuances of both A and B, but this profile makes AB carriers somewhat more likely to develop cognitive impairments and certain infections. However, AB plasma's universal compatibility gives this rare group high value in critical transfusion scenarios. Blood type even influences cardiovascular and metabolic risk. Type O's lower levels of von Willebrand factor mean reduced risk for clot-based strokes and heart attacks, but an increased tendency for excessive bleeding. Types A and AB have higher concentrations of this and related proteins, which may lead to greater cardiovascular and clotting disorder risk. Patterns vary based on region and ancestry, highlighting the intertwining of medical vulnerabilities and inherited traits. Some blood types are so rare they pose unique genetic and clinical challenges. Rh null, missing every Rh antigen, occurs in only a handful of people globally. Managing their transfusions requires specialized blood centers and advanced screening. Patients with sickle cell disease or complex immune profiles may rely on less common variants, making the identification and distribution of rare blood as important as discovery. Blood type also ties to diabetes and metabolic diseases. Type O offers resistance to type 2 diabetes, while A and B are slightly more susceptible, a link discovered through studies of how blood group antigens interface with pathogens and regulate immune response. Continued research into these connections is revealing patterns of risk and advantage that reach far beyond transfusion and into every aspect of health. Ultimately, every blood type marks a unique chapter in human adaptation. They shape how we resist disease, thrive across continents, and survive emergencies. Their frequencies reflect migration, environment, and inheritance, revealing both the strengths and vulnerabilities hidden within our biology. Understanding these hidden advantages of blood type gives an unprecedented look at human diversity, survival, and everyday health.